My pleasure today to. Oh, you wanted to do an announcement quickly.
Errol Akchai, who's visiting from Princeton, and Errol's a theoretician who does modeling of behavior in all kinds of systems, including plant systems. And he's been working with me on plant behavior and its interactions with microbes and their microbial behavior. So if you want to meet with him, he's around this week. Just contact me.
We got any other introductions? So it's my pleasure to introduce Swee Peck. I've gotten to know Swee Peck quite well the last 2 years, seeing as her little daughter Kiri and my son Owen are best buddies. But today she's going to tell us something about both Malayan biogeography and this fascinating ant mutualism system. And so thank you very much for being willing to give it to us, Swee Peck. Swee Peck actually did her undergrad here in Berkeley, and she did a project with Wayne Sosa, and then she did both her PhD and postdoc at Harvard. And this work that she's going to talk about today stems from her time at Harvard.
Well, thanks for the invitation to speak, and thank you all for coming today. The first part of my talk will be on the evolution of host use and ant-plant mutualism, and the second on the historical biogeography of the ants and how that relates to the history of the history of the Sinoland rainforest. So as Rory said, this work was done as part of my PhD thesis, first under Peter Ashton, and then when Peter retired under Nomi Pierce. So the main players in the study system are the ants, plants, and scale insects. The ants live in the hollow stems of the plants, which provide them with food and shelter. The ants protect their host against herbivory, against vines that compete with the plants for light and nutrients, and also possibly against fungal pathogens. The ants also tend scale insects in the hollow stems and harvest their honeydew, an indirect source of food from the plants. For the ants and plants, this is an obligate mutualism in that both partners cannot survive in the wild without each other. The plants belong to the genus Macaranga, and there are about 30 myrmecophytic species, or ant-associated species, in Southeast Asia. The ants are in the genus Chromatogaster, subgenus Deca. There are 22 morphospecies of Cox's scale insect associated with the known to be associated with the ants and plants. And for this talk, I will be focusing mainly on the association between the ants and plants. This is the approximate distribution of obligately ant-associated Macaranga. It closely follows the distribution of ever-wet climate in Southeast Asia. And this is because the association between the ants and plants breaks down in areas where there is a dry season. In this phylogeny of Macaranga, the work of Stuart Davies, my collaborator, the obligately ant-associated taxa are shown in orange, and black denotes the uninhabited plants. The obligate species occur in 3 closely related clades. You can see here's one, and here's another one, and here's the third one. These 2 clades, this one here and this one here, are associated with Deca ants, and this clade is associated with a different subgenus of Chromatogaster. So for the remainder of the talk, I will be focusing on the ants that inhabit only these 2 clades. There are about 22 species here and about 5 species here. And notice also that there have been a few secondary losses of ant association. This is the typical habitat of myrmecophytic Macaranga. They establish at the edges of forests, in forest gaps, and where light and nutrients are plentiful. They are very abundant along logging roads and in secondary or degraded forests. Some species can get very tall, like this one here. And that there is Stuart Davies, my collaborator, who with a Bornean endemic that he recently described. In nature, you never find Deca ants without Macaranga. And without their Deca ants, the plants are attacked by vines and herbivores and eventually die. And here is an example of a plant found without ants, and note the extensive herbivory and heavy infestation by galls. The scale insects, which you can see inside the stems, bunch here, there, are never found outside of myrmecophytic Macaranga. Now there are 2 types of myrmecophytic Macaranga. In this large clade, the stems are naturally hollow. While in this smaller clade, the stems are solid but filled with a soft pith that have to be excavated by the ants, that is easily excavated by the ants. And some species are covered with powdery wax, which experiments have shown are a deterrent to ant species that are not obligately associated with Makaranga. The young leaves of the ant plants sprout nectaries which secrete sugars which the ants harvest. Makaranga is a very large pantropical genus with over 300 species. Many of them form facultative association with ants by providing extrafloral nectar. Stuart Davies et al. Noted that there are differences in the development of extrafloral nectaries in species that are obligately ant-associated versus species that are only facultatively ant-associated. So in the facultative species, the nectaries are disc-shaped and they occur on the mature leaf surfaces. Whereas in the obligate species, the nectaries are cup-shaped and they are active only on the young leaf margins. Another adaptation for ant association is the succulent stipules, which secrete food bodies for the ants. All myrmecophytic Makaranga provide food bodies for the ants. In the majority of species, the food bodies are completely enclosed inside stipules, which makes them accessible only to their resident ants. Here you can see a variety of stipule morphologies of several different species. Note that in all of these, the stipules are completely enclosing the food bodies by being pressed tightly against the stem, kind of like that. And then in these species, the stipules are not pressed to the stem, but they still enclose the food bodies by, so if this is a stipule, they sort of curl their edges under and to form a seam underneath, to form a closed compartments for the food bodies. And in one pair of sister species, the stipules are very small and do not produce food bodies, which are instead secreted by young leaves. I don't know if you can see, but there's some white dots here. Interestingly, these two species are also the most waxy of all the ant-associated species. So the stipule morphologies you have just seen in the previous two slides are from these two clades here. This one here, and this one, which is really not included in my study. Now in this clade, the stipules are open, like you can see here, and the food bodies are exposed. So going back to that large myrmecophytic clade, here is one species from that clade which has lost obligate ant association because the region in which it occurs, the island of Java, now experiences a dry season. So it no longer produces food bodies, and note how the stipules are open and not succulent, unlike the ancestral condition here. The correlation between food body presentation and waxy stems was so striking as to inspire a study by Walter Federley. He showed using phylogenetically independent contrast that there is indeed a correlation. Generally, the waxy species have food bodies that are exposed, and the non-waxy plants hide food from intruder ants in favor of their mutualistic ants. The plants provide three sources of food for the ants: lipid-rich food bodies and sugar-rich exudates from two sources: extrafloral nectaries and indirectly bioscale insects. So in summary, the plant adaptations for obligate ant association are hollow or easily hollowed stems which provide nesting space, food bodies secreted by stipules or young leaves, leaf margin nectaries, and in some species, waxy stems which exclude non-mutualistic ants, and the hiding of food bodies in some species. The subgenus Deca can be identified by counting the number of antennal segments in females. The diagnostic number is 10, and their known distribution is in Southeast Asia, Taiwan and New Guinea, Africa and Madagascar. In Sundaland, in Borneo, Malay and Sumatra, they are restricted to Makaranga. Morphologically, they are a very challenging group to work on, and among the inhabitants of Makaranga, there are 3 published names, but others recognize 8 queen-base morphospecies. The ant adaptations for living in Makaranga include a small crop, which means they cannot store food and are dependent on the plant for continuous food production. This is why obligately ant-associated Makaranga are never found in areas with a dry season. In the dry season, plants do not produce new leaves, and no new leaves means no new stipules, and no new stipules or leaves means no food production. The ants patrol young leaves and remove caterpillar eggs, and they prune away vines which take away light and nutrients from their hosts, and which also serve as a bridge for intruders. Studies suggest that the ants may also be protecting their hosts against fungal pathogens. They have specialized feet and mode of locomotion for climbing waxy stems. And finally, when dispersing females leave their nest in search of new plants to colonize, they use chemical cues to distinguish between host plant species. Experiments have shown that ants on non-waxy hosts are more aggressive and prune encroaching vegetation more, suggesting that ants are more vulnerable to invasion without waxy stems. Experiments have also shown that waxy stems filter out non-mutualistic ants. Ants from non-waxy hosts are able to walk on waxy stems better than ants from non-waxy hosts. And conversely, ants from non-waxy hosts are better able to attach to smooth surfaces than ants from waxy hosts. And this raises the question of whether stem texture might promote host specificity among Deca. When a new seedling is colonized by multiple queens, they fight to the death until there is only one. In the wild, there's always way more new queens than seedlings available for colonization. So competition is very intense. In this young shoot, I counted 17 holes, which means there are 17 queens duking it out inside. And here you can see 2 queens fighting, and this queen is so desperate that she's colonizing the petiole rather than the stem. So I told you earlier that there were only 3 published names among the Deca ants, and they inhabit 27 species of Makaranga. Given that there are that many host plant species, and quite a few of them are endemic to specific islands, it seems very likely that we were grossly underestimating the number of ant species associated with this radiation of myrmecophytes. So as soon as Stuart Davies published the phylogeny of Makaranga, the race was on to publish the phylogeny of the ants. And to answer the burning question, did Makaranga and Chromatogaster co-speciate? Would we see pairwise co-speciation in the ants and plants like we see in this famous example of figs and fig wasps? Or is this a case of ecological fitting, where the ants and plants came together only in recent times and just happened to be a good fit for each other, and coevolution had little to do with the association? A Japanese group was the first to publish a mitochondrial DNA phylogeny of the ants. Based on a sample of 47 colonies, they found evidence for co-speciation between Makaranga and their ants. A couple of years later, a German lab published another mitochondrial DNA phylogeny based on a sample of 33 colonies, and their analysis showed that there was no co-speciation. They found instead extensive host switching. So given the controversy and small sampling of the previous studies, I wanted to do a proper study and settle the matter once and for all. Did Makaranga and their ants co-speciate? But before answering this question, it was first necessary to answer some basic questions. Are the Makaranga inhabiting ants Deca monophyletic? How many taxonomic entities are there? And what is the extent of host specificity among the ants? So to answer these questions, it was necessary to sample extensively. My collaborators also contributed many samples, and these pictures show some of my collecting locations and some of the local assistants. Luckily for me, ant-associated Makaranga, where they do occur, are very abundant. You only need to find a logging road, a secondary forest, or go to the edge of a forest, and they will be there, often by the dozens. There are also Deca in the Philippines and Sulawesi. In these islands, they occur on a different ant plant in the genus Neogene, which you can see here. Swollen internodes with lots of holes. And I was only able to get samples from North Sulawesi. So altogether, my samples include over 400 colonies from 25 of the 27 ant-inhabited Makaranga species in 37 locations. I also included the DNA sequences from the previous 2 studies. To determine monophyly, I included Deca from Sulawesi, Africa, and Madagascar, as well as a variety of other Chromatogaster species. The data comprise CO1 and CO2 mitochondrial genes. The resulting phylogeny shows quite convincingly that the Deca on Makaranga, which is in red, denoted by this node here, is monophyletic. Here's the Asian Deca and the subgenus Deca is probably monophyletic as well. I determined that there were 17 mitochondrial lineages among my samples from Makaranga. I did this by delineating them based on relatively long subtending branches on the phylogram. So for example, in this clade, has a long subtending branch, so I give that an identifying code. To determine whether these lineages were host-specific, I ran chi-square tests to see whether the ants were choosing their host species over the other species in their immediate neighborhood. I found that most mitochondrial DNA lineages show statistically significant host specificity at some host taxonomic level. On the left is the ant phylogeny, and on the right is the simplified plant phylogeny. The asterisk along this column denote lineages that show host preference. The colored circles on the ant phylogeny show the host clade they inhabit, where the color is matched to the plant phylogeny on the right. So for example, these 3 lineages specialize on this clade here. A few lineages, such as these 2, H and K, use a wide variety of hosts but still show preference for certain types of hosts. So given that we found host specificity in most of the lineages, I was then able to test for co-speciation. Using the program Parafit, which randomizes the ant and plant topologies to create a null distribution for comparison, I was able to reject the hypothesis of pairwise co-speciation. But now if we simplify things and take the stem traits of the plants and map them onto the ant phylogeny, we see an evolutionary sequence of events. So note here that the white pie sections are for solid stems that have to be excavated, and the black and gray circles are for stems that are naturally hollow. Black is for waxy stems, and gray is for non-waxy stems. So we see here that the basal-most ant lineages almost exclusively inhabit naturally hollow stems that are waxy. Among the other lineages, the black solid pies, all black pies, are absent. That is, there is none that exclusively inhabits waxy, naturally hollow stems. Instead, we see some species that specialize on non-waxy stems, such as, we see some species that specialize on non-waxy stems, yes, and then some, such as this one and this one, that prefer to use excavated stems that have to be excavated. And some show no preference. So we can see that the colonization of naturally hollow stems, the gray and black, was ancestral, with the ability to excavate stems appearing only within this clade here, where the white pie charts are. And it was the waxy naturally hollow stems that were ancestrally colonized. And then later on, 2 lineages, here and this one here and this one, evolved the ability to colonize non-waxy stems. And now if you put the 2 phylogenies side by side, you'll see that the order of stem texture in the ant phylogeny is similar to that in the Makaranga phylogeny. Within the plant clade that has naturally hollow stems, which is this clade here, waxy is the ancestral condition, just as it is in the ant phylogeny. So we can also reconstruct the evolutionary sequence of ant acquisition in Makaranga, in the Makaranga phylogeny, using the ant phylogeny. The ant tree reveals that the association originated with section pachystemen, and then and sometime later the ants expanded onto a second clade of Makaranga, section pruinosi here, with the stems that have to be excavated. In 1974, Jansen hypothesized that by virtue of their proximity to myrmecophytic species, closely related but non-myrmecophytic swollen thorn acacia species of Central America were induced to evolve ant association because they co-occur alongside their myrmecophytic cousins. Like the acacia, these two groups of Makaranga plants can be found side by side. And we know that competition among queens for host plants is very intense. And we know that some queens will sometimes colonize the wrong species. So it's very likely that, that this clade of Makaranga here provides an example of Jansen's evolution of myrmecophytism by proxy. So to answer the questions I posed earlier, the Deca from Makaranga are monophyletic. There are 17 mitochondrial lineages. Most of these lineages exhibit host specificity, which suggests they might be good proxies for species. The ants and plants did not co-speciation, but their association is not explained by Jansen's ecological fitting, because similar evolutionary sequence of stem texture in the ant and plant phylogenies show that they diversified contemporaneously or co-diversified. And this wraps up the first part of the talk, but I will add that my collaborators in Japan are now working on building a nuclear DNA phylogeny for the ants, so hopefully we will have a proper species phylogeny soon. Now we move on to the historical biogeography of the ants. This region is known as the Sunda Shelf, comprising Borneo, Malay, Sumatra, and Java. Today, the year-round wet climate in the Sunda Shelf supports evergreen rainforests. At the last glacial maximum about 18,000 years ago, Sundaland looked like this. The climate was cooler and drier, with many areas experiencing only seasonal rain. The wet forests that are present now were fragmented, with drier vegetation types replacing them. And we know from the pollen record that savanna vegetation and monsoon or deciduous forests occurred in some places, and there were even hippos on the Malay Peninsula. There have been about 12 glacial cycles in the past 1 million years. During these glacial phases, the continental ice sheets advanced, lowering sea levels and exposing more of the Sunda Shelf, like this. And the climate during the glacial phases was cooler and drier than during the interglacials, and the wet forest became reduced to pockets of refugia while dry forest and savanna vegetation expanded. And here is a reconstruction of what Sundaland might have looked like over the last 12 glacial cycles in the last 1 million years. Again, here's Borneo, Malay, and Sumatra. The questions I want to address are the Deca species a product of cyclic Pleistocene environmental changes? How old are the Makaranga ants of Sundaland? Where did they originate and subsequently diversify, and where were their refugia located? I was drawn to the ant Makaranga system because I've always been fascinated by the rainforests of the Malay Archipelago, having grown up in the region. Because the distribution of these ants follows exactly the distribution of ever-wet forests, they should be able to shed some light on the history of the rainforest, particularly in the area of molecular dating. If you've ever worked on the molecular systematics of tropical trees, tropical forest trees, you will know that a reliable molecular clock does not exist. At the very least, the ants can tell us how long myrmecophytic Makaranga have been in existence. CO1, one of the genes I used to build the ant phylogeny, is reputed to have a clock-like mutation rate. I wanted to confirm this, so I searched the primary literature for arthropod papers with dated CO1 phylogenies that had been calibrated using geologic information. I then independently calculated uncorrected pairwise divergence rates for each of the groups and found that the rate of pairwise divergence seems to wobble around 1.5% per million years for divergences ranging between 3.5 and 10%. So I can use this rate to estimate the time frame for the diversification of the ants. I subjected the CO1 maximum likelihood phylogeny to non-parametric rate smoothing to get a chronogram where the branch lengths are proportional to time. I chose a well-supported node and fixed its age using that 1.5% rate to derive the ages for the other nodes. I repeated this with 2 other well-supported nodes. So here are the 3 nodes. And what I get is 3 timelines, which then give me a somewhat conservative range of ages. And here are the 3 estimated time frames. And the result I got is that the ants are estimated to be 16 to 23 million years old. Fossil pollen data suggests that the current ever-wet forest on the Sunda Shelf can be traced back to about 20 million years ago, after which there is a temporal break in continuity. This suggests that the third time frame is unlikely. I'll set out, and we get a date of 16 to 20 million years for the age of the ants. Now, the similar evolutionary sequence of stem texture on both ant and plant phylogenies indicated that they diversified contemporaneously. So we can also say that the Myrmecophytic Macaranga originated about 16 to 20 million years ago, and this is the age of mutualism. We can also estimate the age of these later evolving Myrmecophytes. They are restricted to this clade of ants here, which corresponds to node 5 here, which takes us to the early Pliocene. So these plants are about 4 to 5 million years old. This blue area denotes roughly the last 1 million years, at least in the top timeline, when the glacial-interglacial cycling began. And you can see that by the end of the Pliocene, which is the gray shading in all the 3 timelines, all of the lineages were already in place. There were no speciation or cladogenetic events in the Pleistocene. The sisters, the Sundaland Decacrema, are in Sulawesi and probably also in the Philippines. And the oldest lineage in the N group is found in eastern part of Borneo, suggesting an origin in or near Borneo and possibly eastern Borneo. These 2 phylogenies show the geographic distribution of the ant lineages and the plant species. Brown denotes Borneo, blue is Sumatra, and pink is Malaya. The ant phylogeny shows a Bornean backbone most of the way, indicating that their center of diversification remained in Borneo. The plant phylogeny shows a similar trend, which, with most of the species being restricted to Borneo, several species distributed throughout Sundaland, and only a few species absent from Borneo. So the center of diversification for the ants and plants remained in or near Borneo. What do I mean when I say near Borneo? In the early Miocene, Borneo did not exist as an island. The emergent parts of the Sunda Shelf look like this. This kind of icky, greenish brown area denotes land, and red is volcanoes. The emergent, so the association could have originated in a place that is today underwater. Note that at this time, Sumatra is only a chain of volcanoes to the west. 5 million years later, Borneo is still part of the mainland, and Sumatra is still a chain of volcanoes. Another 5 million years later, in the late Miocene, the chain of volcanoes begin to coalesce, and now there's a sliver of land connecting it with the rest of Sundaland. By the early Pliocene, this land connection to Sumatra becomes more substantial, and Sumatra has gained some land area. So my inferred timeline for the diversification of Decacrema is consistent with this scenario. There is an old Malayan lineage here, which dates to the mid-Miocene, and we know that Malay and Borneo were connected at that time, but there are no Sumatran lineages older than the Pliocene. So this gives me confidence that the time frame I have inferred is not way off. This curve reflects a cooling trend in climate starting from the mid-Miocene. Mid-Miocene is here. And during that time, the climate was generally warm and wet, with the dominance of wet forests and occasional short-lived appearance of savanna vegetation or dry vegetation in Sundaland. Note that the cooling becomes accelerated in the Pliocene, and savanna vegetation becomes more prevalent, especially during periods of low sea levels. It is during the acceleration of climatic cooling in the Pliocene that we see the cladogenetic events that led to most of the lineages present today, which is this greenish swath here. A good guess is that the cooler and drier Pliocene climate fragmented the ever-wet forest and led to speciation of both the ants and plants. I also suspect that the fragmenting of the forest, and thus the decrease in availability of host plants, was responsible for the origin of ant colonization in the second clade of Myrmecophytes, which, by the ants' estimate, dates to the early Pliocene. A dominant pattern in the biogeography of the ants is a Bornean versus non-Bornean distribution. The colored lines show the distribution of the lineages as reflected by my collecting bias. And note that there is only one lineage, the blue one here, which occurs both inside and outside of Borneo. Also in Borneo, a higher proportion of the lineages are endemic compared to Sumatra and Malaya. These pie charts show the lineages present in each location. I found that the highest diversity location in each of the three islands occur on high elevations on mountain ranges. So in Malaya, the highest diversity site was Cameron Highlands, here on the central range, with 4 lineages. In Sumatra, it occurs on the Bukit Barisan range with 5 lineages. And in Borneo, it occurs on the Crocker range with 7 lineages. This site is close to Mount Kinabalu, the highest peak in Southeast Asia west of New Guinea. Southern Borneo is generally depauperate except for the Maratus Mountains, which have 5 lineages here. I noticed a curious phenomenon in the ant phylogeny. There are 3 pairs of sister taxa with disjunct distributions on either side of the South China Sea. So these lineages in north of Borneo are more closely related to things in Malaya and Sumatra than they are to other ants in Borneo. And these divergences date to the Pliocene between 2 and 5 million years ago. Within this blue lineage, there are 2 migrations out of Borneo. By examining the haplotype network, I found that the haplotypes in each of these 2 locations were each monophyletic. So the haplotypes in Malaya are this one here, and the haplotypes of Lingga are this one here. So this one is, the Lingga haplotypes are more closely related to things in western Borneo, and the ones on the Malaya are more closely related to haplotypes in the Crocker range. And these divergences date to about 1 million, to within the last 1 million years. The real pocket flora refers to a recurring pattern of distribution seen in many plant taxa. It is curiously disjunct and is named after the Riau Islands here. This phenomenon was first noted by Corner in the late 60s and then further expanded by Peter Ashton, my PhD supervisor, first PhD supervisor. The Riau pocket flora was thought to represent wet forest refugia during Pleistocene glacial intervals. These arrows show the presumed dispersal path of the coastal taxa of the Riau pocket with rising sea levels. So if the present distribution of the Riau pocket flora reflects Pleistocene processes, we should expect those floristic communities to date to within the last 1 million years or so. Others are more convinced that the Riau pocket has a much older history than just the Pleistocene. Unfortunately, it's notoriously difficult to date phylogenies of tropical trees because there is no reliable molecular clock for them. So up till now, we had no way of inferring the age of the Riau pocket elements. It's therefore very exciting that these disjunct distributions in the ants look a lot like the Riau pocket distributions. And if they do indeed reflect the Riau pocket, their ages which span the Pliocene through to the Pleistocene suggest that Riau pocket indeed has a much older history than previously thought. We know that the center of diversification for the ants occurred in Borneo, and we can try to reconstruct the origins of the Deca in Malaya and Sumatra using ancestral area cladograms, haplotype networks, and by calculating within location genetic diversity for each of the lineages. There are 6 Deca lineages in Malaya. 3 got there via Sumatra. One was a Pleistocene immigrant from Borneo, and this one here is very ancient and highly endangered, where I only found 2 colonies, and it probably has been there since the mid-Miocene. It appears to be very host-specific, and its host is also rare and highly endangered. There are 5 Deca lineages in Sumatra. 2 are nested within Bornean or ancestrally Bornean clades, and 1 has come from Malaya. The origins of the other 2 are ambiguous. These graphs show the altitudinal distributions of the ants in Borneo and Malaya. Altitude in 100 meters is on the bottom axis. So I noticed an interesting difference between the 2. In Borneo, the ant lineages are both generalists and specialists, whereas in Malaya there are no specialists. But they appear to partition altitude somewhere around 800 meters. I could not include Sumatra because I did not have samples above 1,000 meters there. What could explain this? In Malaya, mountain building activity had ceased before the Paleocene, and there has been little geomorphic activity through the Neogene. Borneo has been tectonically and geomorphically active through the Neogene. There have been volcanoes, notice the red triangles, a plate margin abutting the northern part, lots of orogenic activity followed by rapid erosion at the end of the Miocene. Mount Kinabalu, the highest peak in Southeast Asia west of New Guinea, occurs in the north of Borneo and has been uplifting and eroding ever since the early Pliocene. Although tectonic and most volcanic activity on Borneo has ceased, it has remained geomorphically dynamic to the present. So the historically dynamic nature of Borneo encouraged adaptation to rapidly changing altitudes, hence the presence of generalists, while in Malaya, the older and more stable mountains favored specialization. During the Pleistocene, several lineages underwent sudden demographic expansions, which can be detected by mismatched distributions and Flu's F and Tajima's D tests. The older lineages show a more stable historical demography, and sudden demographic expansions were restricted to 4 of the younger lineages. And their centers of expansion were inferred to be in these locations. The demographic expansions were dated to within the last 1 million years or so, using Mycia 1.5% per million years. So, to wrap up the historical biogeography of the ants, they have their origins in the early Miocene in or near Borneo, probably eastern Borneo around 16 to 20 million years ago. During the Miocene, they continued to diversify in or near Borneo, and there was a colonization of Malaya. During the Pliocene, there was a proliferation of extant species, and continued colonization of Malaya and Sumatra, possibly reflecting the Ria pocket of flora. And during the Pleistocene, there were demographic expansions in some of the lineages and migrations into Malaya and Ria from Borneo. And I think it's reasonable to say that this scenario also probably describes the history of Makaranga and possibly other rainforest species. And with that, I would like to end my talk and thank my collaborators, Stuart Davies, Peter Ashton, collaborators in Japan, Ichino, Shohei Ueda, and my PhD supervisor, Naomi Pierce. And I'll be happy to take questions.
Since the forest trees, moringa, are edge species, what she said, with all the deforestation that's going on there, what is the projection for what's going to happen to these distributions in the future? Wouldn't they, I would think the trees would be expanding.
Yes, they are to a certain extent, but some of the deforestation is so complete that you know, and also you do need good primary forest around for the Makaranga to establish for some reason. So you can't just go to a crappy, totally degraded forest. You won't find them there. There's got to be a source, somehow a source of immigrants good good primary forest for them to continue. Yes.
I was interested in your timescale matching, saying that the tectonic activity of Borneo would lead to more generalization in the altitudinal distribution of the ants, because it seems the ants could move faster than mountains, even in a tectonic sense, you know. That, are these ants poor dispersers, or do you think the ants actually stay put and would actually experience tectonic uplift?
Well, it's about the adaptation, right? So
But I'm thinking the ants, if they want a different altitude, they could just move, right? Or if they wanted to specialize in Borneo, they could find their altitude by moving.
So there is one lineage of ants that is a high altitude specialist. Yeah, I don't know how to answer that question.
It's an intriguing timescales, because I think Dave Wake's salamanders are apparently tracking terrain, so I've heard. So if I just was wondering if it's possible the ants could be equally sedentary, but it still seems a very strange scale matching to invoke tectonics in altitudinal.
and you know, it just might be purely coincidental. It might have nothing to do with the tectonics. But one thing did occur to me that I haven't really talked about with anyone is that these are mitochondrial lineages, and the mitochondria it has to do with energy, sort of metabolism. So you know, it might be reflecting the temperature differences in the altitudes. Yes.
Swee Peck, I was curious about that one lineage that you demarcated in black that's both rare itself and its host plant is rare.
Yeah.
It just seems, given the amount of switching that happens I know it's at the base of the phylogeny rather than the top, that it's a incredibly risky life history strategy for that particular species. Is there something about why it's locked into that host, or?
I don't know, and it's very possible that it's completely extinct now, because I collected it 15 years ago in an area that, you know, just one hill that's surrounded by the city. So it's got very little chance. The host plant is a little bit more common than the ant itself, at least by, you know, like as reflected by the collecting. So yes.
My paleontological prejudice, of course, is that species are always turning over. So you're seeing some relic from a past time that might have been quite diverse. Right, so you know, species longevities are in the order of 3, 4, 5 million years. I don't know about plants, but for animals, right? So if we were to go back to 20 million years, there'd be virtually nothing alive then that is alive today. So at least I don't know anything about this system, but there's always a constant turnover of individual lineages and species.
And I do notice in my phylogeny that the
I don't know that.
the earlier branching lineages.
I don't know about the plants, but I know that from the fossil record. I mean, I look at almost every lineage that I look at, the species longevity is between about 2 and 8 million years. That's just the evidence from the fossil record.
But they would have descendants that might be present.
Yes, but a lot of the lineages are actually extinct. So it's something which Tiago's My Tree paper was about. People don't recognise there's a constant turnover going on all the time, shuffling in and out of lineages. That's just the way the fossil record is. So I'm sorry, I can't help it.
I was more wondering whether it's that it's just losing out as a competitor.
I'm sure it is. I'm sure it's.
For some reason, it's just a poor competitor, given the way these are such aggressive colonizers.
But it might have belonged to a clade 15 million years ago, where it was one of the norms. So it's probably going. But you know, in other words, it's losing now. I would agree, but it might have been once sort of the norm.
And also, this is only a mitochondrial DNA phylogeny, so it might actually be part of a more widespread species that, you know, somehow has captured an old mitochondrial lineage,
But
which
mitochondria
lives
doesn't
on.
lie that much.
No, but yes, I've heard enough criticism that I'm not willing to wager that these are species yet.
You mentioned a couple times that there's no, well, I guess, molecular clock for trees, but there are a lot of really good fossil calibrated phylogenies for tree species that are also in these same sort of communities. So I wondered if you could sort of compare what the insights are in those other studies to maybe co-occurring lineages. For example, Ursaceae, Malpighiaceae, I mean, those are all common elements of many tropical forests. So you probably have a lot of those species in there, same areas.
Yeah, well, okay, so fess up here. I'll fess up here. This work was completed in 2007, and I haven't really been caught up with the. I know that there's been a lot of like Christic and I can't remember who else. They've come up with some.
Well, Kathleen
well-
Kay has an ITS tree.
Who?
Kathleen Kay, who was a postdoc here, she did a paper on ITS. It's a molecular clock. But anyway, I mean,
Yeah, I know. I would
there are
love
lots
to
of
have
plant
the time.
fossils. I mean, in the same region, there's fossils.
There's putatively a Makaranga
Yeah.
fossil as well, but Stuart Davies disputes that.
Oh yeah.
Yeah. No, I would love to have the time to catch up with all this new literature. Hands seem to be tied at the moment.
So that ant lineage that is specialized in dwelling and making the holes in the domain of the trees, any morphological adaptation or like big mandibles or something like that in those?
Yeah, I'm sure there are, and actually, Steph has done the morphospecies, and yeah, I'm sure there are. I mean, and there's one. I think that the earlier branching clades have smaller eyes, and the younger ones have larger eyes, and I'm sure they must have stronger mandibles too. Yes?
Is there also a difference in the level of aggression when you were collecting them? Did you see the ones that are more specialized to be, do you find them to be more aggressive in general, or? Because I know you said in general that there was differences in the types of host.
Yes, I didn't notice, well, I did notice that some were more aggressive than others, but a Japanese group has actually done some of the work. Where is that? And they showed that the plants from the non-waxy hosts are more aggressive, and they prune, they're more vigilant about pruning vegetation. And you know, they sort of counted bites on their hands. But you know, but luckily these don't sting; they just bite. So.
Okay, everyone, let's stop it there. Thanks very much.